In this video, we'll attempt to explain how blood typing works and how blood transfusions work, successful blood transfusions, who can give to who and who can receive from whom. And so we'll start by making some boxes. And since there are 8 blood types in the ABO blood typing system, we'll make 8 rows and just a couple of columns. Okay, so 8 rows. And there's 4. And there is 8. And we could just maybe do something like that. All right. So then we can start by writing down over here the 8 different blood types. So we'll put A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, and O negative. Now, the next thing we should do is get some definitions down, or just sort of explain some terms that will come up. For example, what are antigens? What are antibodies? What is agglutination? And so let's just put that over here. Antigens. What are antigens? What are antibodies? So antigens on red blood cells are proteins that are self-identifying proteins. They're like the flags to say, hey, this is what I am. So we can just say self-identifying markers. And this is how we know who is who, which blood type is which, by these antigens. You'll find them on the surface of the red blood cell. All right, and so for the next, antibodies. What are antibodies? These are also proteins that are floating around in our entire body, but specifically in the blood, they act as part of our immune system. And what they do is they capture foreign things, pathogens, foreign invaders, and such. Anything that doesn't belong, they capture and clump together and wait for macrophages and such to come in and phagocytize, destroy the foreign molecules. They don't really attack. It's more like they grab on and wait for other molecules to come and attack. But we can just call it, for our purposes here, we can just call it the attackers, part of the immune system, just to make it easy. And so we'll see these examples as we fill this chart out, or this table out. Okay, so if we are to start drawing out a whole bunch of antigens and antibodies, we need to make a key. And so we need to make some kind of shape for the A antigen and some kind of shape for the B antigen. Although proteins really do have shapes, they don't necessarily have the a circle or a square or a triangle. But for our purposes, let's make the A antigen circle and the B antigen triangle. So here we go. So here is a flag circle, and we'll call this the A antigen. There's our A antigen. And let's draw out the B antigen. We'll make that a triangle. There's our little triangle flag. And we'll call this the B antigen. So there we have it. We have our two shapes. We got the circle here. I'll make that a little bit bigger so you can see it. And we got our B antigen. We'll make it a triangle. And then the next thing is, well, if this is the shape of the A antigen, then what do A antibodies look like? What do B antibodies look like? Well, let's say one of our antibodies, they look like this, or the basic legs. If the job of the A antibody is to attack foreign invaders that don't belong, they're going to have the exact opposite shape. So if this is a circle, the A antibody then will have the concave version of the circle. And so here you have 3 spots where the A antibody can bind to 3 different A antigens. So here you go. This is an example of an A antibody. And it's important to keep those 2 words straight, antigen and antibody, because they are really, really 2 different things. And here, this shape, it would fit into the triangle. So these are the triangle antibodies, and these are the circle antibodies. So if this B antibody is floating around in the blood, it's going to be able to attach to only this type of shape, which is, since this is the opposite or the concave version of the triangle, see the triangle could just fit right in there, then this will be essentially looking for triangle shapes, which would be the B antigens. And since this is a circular shape, this antibody will be floating around and be able to bind to anything foreign, which should be the A antigens. All right, so let's draw what someone with A positive blood look like. So if we put a couple of, let's say we'll just put in 1 red blood cell. What the question is, how do we depict that this is a red blood cell from someone with type A positive blood? And the answer is, well, remember, these antigens are the self-identifying markers, and so that's the only way that we can show that this person is A positive. So all we got to do is put a bunch of these flags on, and there we have it. So here are the flags. You need at least one, but you can put on a whole bunch if you want, and there are our flags. These are the antigens. They're circular because that's what our key, original key, shows. And we can do the same thing with this one. There you go. Now, and let's ignore this positive and negative for the moment and just continue. So we know now that we have successfully depicted that these two red blood cells are a person with type A blood. Why? Because we have put on the red blood cell self-identifying markers. These are the antigens, and more specifically, A antigens. Now we do the same thing again with type B blood. We got to put, how can I show that this is a B red blood cell? You put on B antigens. Triangle, triangle, because that's what our key shows. All right, and AB, what does that mean? Is there such a thing as AB antigens? No, there are A antigens and there are B antigens. There's no such thing as an AB antigen. This is just showing that there are two different antigens. And so you put on maybe even a few more flags. There are A and there are B on this red blood cell. Just like that. And finally, what kind of antigen is the O antigen? No such thing. There is no such thing as an O antigen. So what does the O mean? It means that there are no antigens. Or at least there aren't any A's and there aren't any B antigens. And so you just leave these just like that. Since there aren't any circles and there aren't any triangles, because this person has type O, meaning there are no A and no B on this person's antigens on this person's blood. Now let's go back up to the top and let's get this positive and negative thing down. What does this mean? This positive negative? This is depicting what a third antigen, which is the Rh antigen. The Rh antigen is another antigen that we have to be aware of. And if the person is A positive, that means they have that Rh antigen. And if they're A negative, they don't have that Rh antigen. So then since this person is A positive, let's add on that antigen, at least one, to show that. And sometimes the Rh, another letter for Rh is D, is the letter D. You can say D antigen or Rh antigen. And so if you add that on to this person, now you have successfully shown all the antigens that are attached to this person's red blood cells. Should we put attach a D flag onto this A negative person? No, because negative means that they are missing that antigen. So no, we just leave it just as it is. And then you just continue all the way down. The B positive has a D antigen. And we leave the B negative alone. The AB positive has a D antigen, and you leave the AB negative alone. The O positive person does have the Rh antigen, the D antigen. and the O negative person does not have the D antigen. That's what the positive and negative means. Do you or do you not have that third antigen called the Rh antigen? Rh coming from rhesus monkeys. All right, now if we go back up to the top, let's ask one question for each of these different scenarios. A person with A person with A positive blood, how many antigens does this person have? How many different types of antigens? And the answer is two different types of antigens. They have the A antigen and the D antigen. So there's two different types. A person with A negative blood, how many different types of antigens? Answer, one. Just one kind, which is the A. How many different types of antigens does a person with B positive have? Answer, two. One, two. Two different kinds. Here, B negative, one. Just one kind, which is the B. How about a person with AB positive blood? How many different kinds of antigens? Well, you got the circles, you got the triangles, and you have the D. There's three different kinds. And how about a person with AB negative blood? Two. One, two. And finally, a person with O positive blood will have how many different kinds of antigens? Just one, which is that D antigen. And how many different kinds of antigens? Well, they don't have any antigens, the O negative, because they don't have A or B or D because they're negative. All right. That concludes how to make a table and how to lay out the foundation for blood figuring out who can receive from who and who can give to who, the donors and the recipients. Now, generally when you give, when there is blood given, donated, it's just the red blood cells that are given. Just these. Not just the red blood cells, which contain the antigens, not the antibodies. When you ask now the next question to ask yourself is, what else is going on in the blood? What else do you have in the blood concerning the blood typing system? Well, we also have antibodies. We have to put these in. And so there's a question or something that you can commit to memory to know who has which kind of antibodies. And that is, so put like antibodies question mark, and the answer is who had, what antibodies should I place in all of these different scenarios? And the answer is all other shapes. All other shapes. But we will ignore the D for now. So we're just going to deal with the circles and the triangles, the A antigen and the B antigen. So a person with A type A blood has what shape antigen? And the answer is circle. And how do we know what shapes to put for antibodies, or what kind of antibodies do they have? The answer is all other shapes. Well, if you have circle, then then this person is gonna have triangle antibodies, the B antibodies. So this person definitely has triangle shape antibodies, B antibodies. There you have it. And this person, same thing, circle. So they're going to have all other shapes, which is the triangle. And we're going to ignore this D antigen for the moment. a person with B blood has triangular antigens. And what kind of antibodies should I put in here? Go back to our little code here, and the answer is all other shapes. If this person has triangle antigens, they must have circular antibodies. Circular antibodies. Just like that. Now we got AB blood here. What type of shapes antigens do they have? They have triangle and circular. And we need to put in all other shapes. Well, there's only 2 shapes involved. If we ignore the D, this person has triangle and circular. So there are no shapes left over. So no shape, no antibodies. No antibodies, because they're both taken up. Again, no antibodies for both AB positive and B negative. And finally, what shapes? Circular and triangular. The person with O positive and O negative doesn't have circular or triangular antigens. Therefore, all other shapes are both shapes. So this person actually has circular and triangular antibodies. A antibodies and B antibodies. What is the function? One more time. What is the function of these antibodies? They float around looking for foreign things. Well, I hope it makes sense that we make sure to put in all other shapes, not the same shapes, but the opposite shapes. Why? Well, because if this person has circular antigens and they had circular antibodies, the circular antibodies would fit into the circular antigens and they would clump. That which is a bad thing, which is essentially attacking. And that is called agglutination. That's called agglutination. And if we are to draw a picture, let's say down here, this is an A antibody. What can this thing do? If you have a red blood cell here, you have a red blood cell here, and here this antibody can bind up to 3 red blood cells. And there are the antigens on so because the shapes fit, this antibody will attack these 3 red blood cells, which is what you don't really want happening all over in your blood. I mean, you don't want your own antibodies attacking your own blood that your body is making. You want your antibodies to attack things that are foreign. If you had circular antibodies normally in your blood, you'd want to be the other shape. That way your own antibodies don't attack your own red blood cells, but other things that would get in to your system. And so I hope that makes sense. Therefore, these antibodies in each of these different scenarios had better be different shapes than the antigens associated with it. If these are triangular, these better be circular. Triangular, circular, circular, triangle, circular, triangular. The red blood cells here have both A and B antigens. What kind of antibodies can you put in here? None, because the triangular ones would bind to the triangles, and the circular antibodies would bind to the circular antigens. That would be a bad thing. You don't want to have A or B antibodies in your blood if you have AB blood, because your own antibodies would be attacking your own red blood cells. And lastly, what type of antibodies do we want to have in O blood? Well, you want to have both, because you don't have any antigens. Now that concludes laying out the groundwork for blood transfusions. Now we ask ourselves, well, why aren't we dealing with the D antigen here, with the Rh antigen? Why don't we have Rh antibodies? Well, the answer is because people aren't generally born with Rh antigens, but we are born with where people aren't born with Rh antibodies, but we are born with it's in our genetic code to make A antibodies and B antibodies, but not Rh antibodies. So when a person gets sick, when a person gets a cold, flu, what have you, the immune system develops antibodies against those particular invaders. So by exposure, you start making antibodies. But these two we are born with. It's in our genetic code. We didn't have to be exposed to anything to have these antibodies. We are born with them, and that's why we are putting them in all of these different scenarios. However, that is the way the D antibody works. You only have it in your blood if you are exposed to that particular antigen. However, only if it's foreign. This person will never produce Rh antibodies because it's not foreign to them, because the D antigen is not foreign. It's part of their blood. But this person might form Rh antibodies. A person with B positive blood will never make, even if they're exposed, will never make Rh antibodies because they have that D antigen anyway. The D antigens are not foreign, so they'll never make Rh antibodies. But a person with B negative blood, if they were exposed, they got someone else's blood into their blood that have the D antigen, they will start making the Rh antibody to attack those foreign Rh antigens. So because some people might get it down the road, might be exposed down the road, and some because we're not generally born with Rh antibodies, we just don't put it in these boxes so that we don't confuse anyone. But it is in our genetic code that we are born with the A antibodies and the B antibodies, even though we weren't exposed to those foreign particles from birth. Now we ask ourselves, well, who can give to who? Who can give to who, and who can receive from who? And the answer, and before we can answer that, we have to understand that when we draw blood, we call that whole blood, and then we'll centrifuge it. It's centrifuged, and we separate the blood into its different constituents. The red blood cells are generally the only thing that you'll give when you're giving blood. You don't give the red blood cell, you don't give the antigens and the antibodies, you only give the antigens or the red blood cells. You won't give both. Otherwise, it would be really, really hard to do any blood transfusions. So, we should put a little note up here that we look at only donors' antigens because we don't care about their antibodies because we're not giving antibodies. And we look at only the recipient's antibodies. Why? Well, because that's the only thing that's going to be doing the attacking. We don't care about the recipient's antigens, really. We don't really care. And because they don't attack. And we don't care about the donor's antibodies because we don't give them in the first place. We only give red blood cells. The antigens are found on the red blood cells. So if we are to take any example here, and actually before we, we can just give a couple of examples. So let's just give an example here. Let's say someone with B positive blood is to donate their blood to someone with AB blood. The question is, can that work or can that not work? So let's put an arrow first just so we can keep track of what we're talking about. And this arrow will show that we're going to give the B positive blood to AB positive blood. Can this work, yes or no? Well, let's look at the donors. This would be the donor here. Let's look at the donors, antigens, and we look at the recipient's antibodies. We're not going to give both of these. We're only giving the red blood cells, the antigens. These are triangular. These are B antigens. If we are to place them into blood with AB positive blood, will this person's, we look at the recipient's antibodies, will this person's antibodies attack antigens with triangular antigens? And the answer is no, for a couple of reasons. For number one, they don't have any antibodies. They don't have any attackers. And therefore, they're safe. There's another reason, there's another way you can look at this. You can ask yourself, is there anything foreign coming into this blood? Well, what does this person have? This person has triangular shape and D shape. Well, a person with AB positive blood also has triangular shape antigens and D shape antigens. There is nothing foreign coming into this blood. This person has everything that this person has. There's nothing foreign coming in, and therefore there are no worries. And therefore this does work. We can put a little smiley face to show yes, this does work. Okay, let's do it again. Let's just come up with another scenario. Let's say B negative to A negative. Does this work? B negative to A negative. Let's ask the question again. We look at only the donor's antigens, and we look at only the recipient's antibodies. This person has triangular antigens. We ignore the antibodies because we're not going to give these anyway. We're only giving the red blood cells. And we look at the recipient's antibodies. Do these two shapes match? This person has B antigens. This person has B antibodies. Do the shapes match? The answer is yes. That would be a bad thing. If we put these red blood cells into this person's blood, this person's antibodies will attack because those shapes fit. Just like we see here. Those shapes would fit, and you would get agglutination, which is a bad thing. This would not work. This person's antibodies would attack all of these red blood cells coming into the body. No, 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 no. Another way you can look at it is there anything foreign coming into this blood? Yes. You got triangles, and this person doesn't have any triangular antigens on their red blood cells, therefore triangles are foreign. I don't want anything foreign coming into my blood. I only can accept the same shapes. Therefore, this scenario would not work. If you want to see actual slides, here's an example of an actual slide of actual blood with that have 3 different antibodies dropped into their blood. So these two look fairly the same, and this one looks different. What we did here is we dropped someone's blood, put a couple of drops here, a couple of drops here, and a couple of drops here. And then we went back and added A antibodies, and we added B antibodies, and we added D antibodies. And this is what you get. You got agglutination here. The red blood cells clumped up so much that they pretty much disappeared. And you have agglutination here. The red blood cells clumped up so much that it's totally clear. You can't even hardly see the red. Where the B, in this B area, it's very smooth and it just looks like regular blood because there was no agglutination. So you ask yourself, okay, well, this person dropped their blood here, here, and here. They added antibodies here, and antibodies here, and antibodies here. And you got agglutination here and here. What does that mean? Well, it means that this person is type A positive. I added antibodies where they shouldn't have been added. I added antibodies into a person's blood where they don't exist, If they did exist, you wouldn't have any agglutination. And that's how we can type blood in the real scenario. All you need to do is take your blood, put it in 3 different areas, then you add the antibodies of those 3 different antibodies and see if they clump up. If they clump up, obviously you don't have those antibodies in your own blood, because then you would get agglutination constantly in your own body. So if it suddenly you see it up, that's what I must be, because I obviously don't have those antibodies in my own blood. This person must be A positive. Let's do another example. Here's another one. And the reason why like the B is backwards is because you take a Sharpie was taken on the back just so that the Sharpie, the actual Sharpie ink wouldn't get mixed, so that this is actually written on the back side of the slide. So here you have real smooth blood. Let's see here. This is also smooth blood here, and here you can see you got some agglutination going on. There's your RH. There's your D antigen and antibody component. So again, what do? You take your blood and you drop it here, here, and here. You add in A antibodies, B antibodies, and RH antibodies. and see if you get any agglutination. If you do, that's bad, and that must mean that you don't have that particular antibody in your blood. Otherwise, that's what you would get all the time, which is not a good thing. And therefore, this person must be, what do you think? What type of blood type? Not here, not here, but here. This person has O positive blood. O positive blood. And that's how you do blood typing and figuring out who can give to who in blood transfusions. Let's do just one more scenario, just to make sure you got it down. And let's give O positive, or let's say let's give someone with B negative blood. Let's give B negative to O negative. B negative to O negative. We only look at the donor's antigens. So we look at the triangles and we want to place it into O negative blood. Can this work? Yes or no? Ask yourself, well, we look at the recipient's antibodies. Circular antibodies and triangular antibodies. The answer is no, this would not work because this person has these two shapes would fit, which is a bad thing. You would have agglutination. This would not work. For all those red blood cells that come into this blood, this person's antibody, this person's specifically B antibodies, will constantly attack until they're all gone. You'd get that agglutination. So we always hear this universal donor and universal recipient. Universal donor and universal recipient. Who is who? Well, this person, it seems like a person with O negative blood can't receive really from anyone because they have so many antibodies. And a person with AB positive blood could receive from anyone because they have no antibodies to attack. Turns out that the O negative, person with O negative, can give to anybody because they have no antigens on their red blood cells. They don't pose a threat to anybody. This person is not foreign to this. There's nothing foreign going into here, nothing foreign going into here, and so on. There's nothing on the red blood cells, so they don't pose a threat to anybody. But they can't receive from anyone except from themselves. O negative to O negative, because they have so many antibodies. Where on the flip side, so O negative must be the universal donor. And on the other side, someone with AB positive blood must be the universal recipient because they have no antibodies that would do any attacking. This person's blood can go in here, no problem. This person's blood, this person's blood, this person's. So AB positive must be the universal recipient. AB negative is very close, except they're missing that D antigen, which would mean if O positive gave to AB negative, that is a foreign antigen. What would occur if we did this? What would occur? This person would start producing D antibodies until these are all gone, because this is foreign to this person. So this doesn't work. Negatives can usually give to negatives, and positives. Negatives can give to negatives and positives sometimes, but someone with a positive can never give to a negative ever, because those recipients will always end up making antibodies against those Rh antigens. So you can never have a positive give to a negative, but you can have a positive, usually have a positive give to a positive, and a negative to negative, and a negative to a positive, but never a positive to a negative. So I want you to since you're going to be tested on this, I want you to go through several scenarios and be able to, well, first off, be able to make this table from scratch without any notes whatsoever, and see if you can replicate what we did here. And then after you've done that, can you go through different scenarios of blood transfusions and see if you can figure out who can give to who and who can receive from who. And that concludes the blood typing section of the chapter.